Welcome back. So we will continue once again. Uh, they transferred the cell into the medium with normal N14 14NH4 Cl and took sample at various definite time interval as the cells multiplied and extracted from the DNA that remained as double standard strand helix. The various samples was separated independently on the CSCL gradient to measure the densities of the DNA. Okay, can you recall that the centrifugal force and think why the molecule with the higher mass density would sediment faster? The result are in this way. We can see in this figure we can see N15 DNA. Okay, and when it goes into generation N15 see blue color is N15. Okay, after the 40 minute it was generated in only blue. Is it? Next one is gravitational force. Due to the gravitational force, and it will be the hybrid, heavy hybrid, light and hybrid. See, blue color refers to the light and dark color refers to the hybrid. And this is which we call as the separation of DNA by the centrifugation. What we call as centrifugation. So next one is third one, the DNA that was extracted from the culture one generation after the transfer from the N15 to N14 medium, that is after 20 minutes. Is it? E. coli divides in 20 minutes, had a hybrid of the intermediate densities. Is it? And DNA extracted from the culture after the another generations, that is after 40 minutes, second generation, composed of the equal amount of this hybrid DNA and of light DNA. If E. coli was allowed to grow for 80 minutes, then what would be the properties of the light and hybrid densities DNA molecule? Is it? So next we will see a very similar experiment involving use of the radioactive thymidine to the detect distribution of the newly synthesized DNA in the chromosomes was performed on Vicia faba. Faba means white Taylor and Collegius in 1958. The experiment proved that the DNA is chromosomes also replicate semi-conservatively. Okay, so next we will see the next subtopic is the machinery and the enzymes. See, okay, the machinery and the enzyme in living cells such as E. coli. The process of the replication requires a set of the catalyst enzyme. The main enzyme is referred to as a DNA dependent DNA polymers. Since it uses a DNA template to catalyze the polymerization of the deoxynucleotides. Is it? These enzymes are highly efficient enzymes as they have to catalyze polymerization of the large number of the nucleotides in a very short time. E. coli that has only 4.6 into 10 to the power 16 BP. Okay, compared with the human whose diploid content is 6.6 into 10 to the power 9 BP. Is it? Completes the process of the replication with the 18 minutes. That means our average rate of the polymerization has to be approximately 2000 BP per second. Not only Do these polymerases have to be fast, but they also have to be catalyst the reaction with high degree of accuracy. Any mistake during the replication would result into mutation. Furthermore, energetically replication is very expensive process. Deoxyribonucleoside nucleoside triphosphate serve dual purpose in reaction. The two terminal phosphates in a deoxynucleoside triphosphate are high energy phosphates, same as case of ATP. Is it? In addition to the DNA dependent DNA polymerases, many additional enzymes are required to complete the process of the replication with a high degree of accuracy. For the long DNA molecules, since the two strands of the DNA cannot be separated in this entire length due to very high energy requirement, the replication occurs within a small opening of the DNA helix, referred to as replication fork. The DNA-dependent DNA polymerases catalyst polymerization only if one direction, that if 5 minutes to 3 minutes. This creates some additional complication at the replication fork. Consequently, on one strand, the template with the polarity 3 minutes to 5 minutes, the replication is continuous, while the other the replicate with template with polarity, it is discontinuous. The discontinuously synthesized fragments are later joined by the enzyme DNA ligase. Okay, figure 5.8. We can see this is the figure. Okay. So next we will see the DNA polymers on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. There is a definite region in E. coli DNA where the replication originates. Such regions are termed as origin of the replication. It is because of the requirement of the origin of the replication that is a piece of the DNA is needed to be propagated during the recombinant DNA procedures requires a vector. The vectors provide the origin of the replication. Further, not every detail of the replication is understood well. In eukaryotes, the replication of DNA takes place S phase of the cell cycle. The replication of the DNA and the cell division cycle should be highly originated. A failure is cell division. Further DNA replication result into ploid, a chromosomal anomaly. You will learn detail in the process of in this higher classes. Thank you for watching.